Welcome to our presentation on folic acid and neural tube defects. We'll start off with a brief overview of the link between folic acid and NTDs. Neural tube defects, or NTDs, result from the failure of the embryonic neural tube to close completely around the 28th day of pregnancy. The two main types are anencephaly, which is when a baby is born without parts of its brain and skull, and spina bifida, which occurs when the backbone does not close properly and affects the spine. While it's difficult to prove causation, the protective benefits of increased amounts of folic acid at periconception and during pregnancy against NTDs has been well established. Research suggests that folate taken before and during pregnancy may also prevent other congenital birth defects such as cleft lip, cleft palate, and neurodevelopmental disorders like autism. Observation rates of NTDs are higher among economically disadvantaged populations with poor diets. This is not surprising since dietary folate is generally found in leafy green vegetables like spinach and citrus fruits. We will now discuss some of the major research studies that examined the relationship between folic acid and neural tube defects. Various study designs have been used to examine this relationship. Variations exist regarding the form of the nutrient and at what point in the pregnancy the nutrient was added or removed. Many of the research studies conducted assessed the use of supplements, with the exception of 4, which assessed folate from dietary intake. Researchers might choose supplements due to their increased bioavailability in comparison to dietary folate. The use of supplements also allows for the distinct start and stop of supplementation. Timing of assessment is important as well. Molunsky and colleagues examined folic acid supplementation nearing the end of pregnancy. They found that the use of supplementation started after the first 6 weeks of pregnancy was unrelated to risk for NTDs. Case control and cohort studies present some interesting findings. Retrospective studies can be difficult due to recall time. Prospective studies also present some issues. For example, blood samples of folic acid are difficult to obtain because of the high level of stability needed for the sample. Also, collection time plays a big role. If samples are collected after the critical point of development in the pregnancy, the recorded levels of folic acid can provide misleading information. Daly and his colleagues conducted a study that indicated a connection between blood and plasma folic acid levels and NTDs. This was a nested case control study which was composed of 84 women who had pregnancies affected by NTDs and matched them threefold against unaffected pregnancies. The blood sample results indicated that the affected women had lower levels of folic acid in their plasma and red blood cells. Meta-analysis showed inverse relationships between blood and plasma folate and NTD risk. This inverse relationship can be seen here in Figure 22. There have also been various randomized and non-randomized clinical trials. The Medical Research Council study was composed of women who had previously been affected by NTDs. They were randomly assigned to one of 3 groups: the supplement group, taking 4 milligrams of folate per day, the placebo multivitamin group, who took a regular multivitamin, and lastly to the placebo group. However, research was halted early due to significant findings that showed the protective effect of folate in the supplement group was 72%. A second study examined women who had not previously been affected by NTDs, and they were randomly assigned to either a supplement group, taking a multivitamin, which had 0.8 milligrams of folic acid, or to a placebo group. This study also ended prematurely due to 6 cases of NTDs which were found in the placebo group. Clearly, one of the biggest issues with these randomized clinical trials is of an ethical nature. Women who aren't taking the folic acid supplements are having higher incidences of NTDs, and so they had to be stopped early. Berry and his colleagues conducted a non-randomized trial in China. The northern region of China had been highly afflicted with cases of NTDs in comparison to rates in the southern region. Women in both geographic areas who had no prior NTDs were instructed to take 0.4 milligrams of folate a day. Results showed an 85% decrease in NTD cases in the northern region, as well as a reduction in the southern region. This drastic drop in the north basically evened out the risk of incidences in both areas. This is a visual representation of that drastic drop and leveling out of the NTDs. So there are limitations to various research studies, including the misclassification of exposure, which can occur if errors are made regarding the time in which vitamin supplementation and testing began, inverse relationships in the conclusions between similar research studies, necessary reliance on participant recall of supplement intake, and with a limited number of cases, which can create a wide confidence interval and decreased precision, it's hard to draw conclusive results. This brings us to some of our biggest questions regarding research findings. First, is folic acid actually responsible for the rates of incidences of NTDs? We are always wary of assuming causality in research studies, so this is a big question. Second, will more or a higher dose of folate prevent the cases that still occur? Basically, how much is too much, how much is not enough, and what is the correct dose? And lastly, are there specific women, perhaps genetically, who are more susceptible to NTDs, and will increased folate help reduce their chances of being affected? After various considerations on how to create policy, there appeared to be three choices. The first was to advise people to increase their intake of foods rich in folate. Fruits and vegetables are the main dietary source of folate, so there are other health benefits to this idea. However, the bioavailability of folate is lower than that of in supplements, and it's variable among food items. Additionally, fruits and vegetables can be more expensive than other food items, thus making them less accessible to low-income populations. Lastly, there is no way to enforce the increased intake, and so you can't ensure folic acid through diet. The second option was to recommend folic acid supplement for women who are of childbearing age. The CDC, or Center for Disease Control, advises any woman who can become pregnant to take 0.4 milligrams of folic acid daily. The cost associated with this approach is less expensive than the whole food approach, but again, there's no way to ensure that all women are following this recommendation. The last option concerns folic acid fortification of a food, like flour, because it would ensure reaching almost the entire population, including women who have unplanned pregnancies. The issue with this approach brings us back to a previous question. How much is too much? How much is enough? And are there any negative health effects associated with fortification for the general public? One such concern exists surrounding vitamin B12. Folic acid can mask or hide the symptoms of anemia resulting from a B12 deficiency. This deficiency can cause serious neurological issues. The population at large consumes folic acid at varying amounts due to a number of factors, so it's important for the policy to select an amount of fortification that would allow for positive health benefits regarding NTDs, but not too high to create other health risks. After taking all of these into consideration, the FDA settled on fortifying 0.14 milligrams of folate per 100 grams of flour, and this was put into effect in 1998. So post-fortification, there is generally positive results. The CDC estimates spina bifida rates declined 25% from 1995 to 2000, and an even further decline of 13% from 2000 to 2005. However, some populations are less susceptible to the fortification of flour. For example, Mexican Americans consume more corn, which is not fortified. Thus, the fortification was not reaching the entire population to its maximum ability. A big question for future policy is, could we fortify our foods more in order to prevent the remaining incidences of NTDs? Further fortification has some benefits. Firstly, folate has been associated with decreased rates of stroke and coronary heart disease. Secondly, research indicates that fortification could lower the incidence of neural tube defects even more than it already has, meaning the dosage could increase and it might reach more cases of NTDs. There are also some possible risks that exist if policy decides to further fortify. The previously mentioned concerns about B12 deficiency masking anemia could be real for some people. Another possible but unlikely risk is an increase in colorectal cancer. The concern arises from some research that indicates folate might aid or accelerate the development of pre-existing tumors. That said, the fortification of folate coincided with a nationwide effort to increase colon screenings, which could explain the greater increase of diagnosed colorectal cancer cases. Folate may not be responsible for the increased rates. It may have just been that more people were being screened for it while fortification was taking place. There are definitely opportunities for further research regarding folic acid and NTDs. One area involves a link between genetics and the dietary interactions. Frost and his colleagues noted a common polymorphism 677 in the 510 MTHFR gene. This gene plays a role in converting amino acids, and it's integral to making proteins within the body. This gene is also associated with a decrease in the folate enzyme activity. Various other studies have similarly noted that the polymorphism might play a role in the occurrence of NTDs. However, more research is needed. Additionally, research using fortified foods with the addition of vitamin B12 is needed to understand the relationship between these nutrients and the risk of cardiovascular disease, cancer, and cognitive function. Thanks so much for taking your time and attention. Let us know if you have any questions regarding folic acid and NTDs.